Hey, what's going on, everybody? This is your host, Sergio Picasso, aka the Custodian and founder of the Connect community. I'm going to be moderating today's panel of this episode of Under the CannaScope, hosted by MyFloraDNA. So first off, I want to thank everybody for tuning in. The purpose of the show is really just to educate. We want to bring the community together. We want to provide access to some worldwide industry experts in subject matter of their respective field, and ultimately continue to grow some fire. So before I jump into that, I just want to go ahead and do a little bit of housekeeping. This is a live stream, so this will be recorded. Be sure that you subscribe to the MyFloraDNA newsletter, which is under the CannaScope. So we're going to be doing a lot more of these educational discussions, bringing on some really great panelists to discuss some great topics. And again, really appreciate you guys tuning in with us. So without further ado, I'm going to go ahead and just go through a quick lineup of what we're going to be going through today, which the topic is integrated cultivation health management. So we're going to be diving into the world of IPM, the world of nutrition. We're going to ultimately get to the point of Q&A. So we want to bring on the panelists. We're going to do a quick bio of each of these subject matter experts, and then we're going to jump right into some key topics around the topic itself. So without further ado, I'm going to go ahead and welcome to the stage Mr. Ajith Anand, CSO of MyFloraDNA. Welcome, Ajith.
Thank you, Sergio. It's really an exciting time to be here with you folks. Those who have not known me, my name is Ajith Anand. I'm the Chief Scientific Officer. I have a PhD in plant molecular biology with an MBA with over 20 years of experience in the industry, not necessarily in the cannabis, but I've been in the seed industry for a long period. MyFloraDNA is a data and science-driven company which provides services and solutions which is sustainable, cost-effective, and benefits our community, especially the cannabis community. We are here to partner and learn, share knowledge from this community so that we can enable our breeders, our growers, and our cultivators to propel the business. And we are, as I told you, we are a really science and data-driven company. And so we believe that we want to provide all the knowledge of the recent advances in science and technology to this community so that they can find purpose and the best way to utilize it for their own business growth. Thank you.
Perfect. Appreciate you. Thank you so much for taking the time today, Ajith. Next to the stage, I'd like to welcome Matthew Gates. Matt, can you hear us okay?
Oh, sorry about that. My name is Matthew Gates. I'm an integrated pest management specialist. I've been working in agriculture for 13 years, and I have a degree in horticultural science. And I'm excited to talk about all of the various topics for IPM and how we can improve people's cultivation strategy.
Amazing. Appreciate you joining us, Matt. And I apologize, guys. I think I might be having a little bit of a network connectivity issue here, so I apologize if I'm on delay here, but I'm going to go ahead and get us on to our next panelist, Mr. Damian Solomon. Welcome, Damian.
Hi, everybody. Glad to be here. So I'm Damian Solomon. I've had 25 years experience in controlled environment agriculture, predominantly high-tech glasshouse and high-tech indoor production facilities. In that time, I've built large-scale greenhouses. I've done large-scale projects, indoor projects. And in the last 10 years or so, I've been focused mainly on cannabis crops, although I still work in food crops. But I have two businesses. One's Plant Geek Consulting, where I do cultivation consulting. And then also my second business is King Solomon Nutrients, where we're a hydroponic nutrition company that services not only cannabis, but global agriculture in terms of controlled environment agriculture. So it's a pleasure to be here. I'm super stoked to hear from some of the other panelists, and I know that we're all working together to help bring knowledge and just help open up the community to empower the community with better data so they can make better decisions. Happy to be here.
Right on, Damien. Appreciate you joining us today, man. Thank you. And last thing, Mr. Kevin Crouch. Welcome, Kevin.
Hello, my name is Kevin Crouch. I have a degree in wildlife biology and practiced marine and wildlife biology for almost 20 years. Did a lot of environmental consulting, and I've been growing cannabis for 20 years and breeding and experimenting for pretty much that whole time. I currently operate a double-decker large LED grow, Delighted, in Sacramento. We have 1,170 flowering lights, and in the current platform, I'm always experimenting, trying to just better the cannabis plant and the community. Thank you for having me.
Appreciate you joining today, Kevin. So, guys, I just want to say to all for taking your time out of this day. Each of us has a unique perspective, history. Ultimately, again, going back to the purpose of this discussion, value in education and awareness, and ultimately about plant diseases, right? So IPM, plant diseases, nutrition, a lot of this stuff ties in together. So we thought this would be a great way to align us all. And I want to jump our first topic about plant nutrition. So Damien, I'm going to pass you the baton, and let's start talking off about how plant nutrition and inputs can be derivative of plant disease.
Yeah, great. I mean, that's a good segue. You know, I mean, I think when you look at terms of plant nutrition and plant health, plants obviously are going to require certain key elements to ensure that their cellular metabolism and all of their processes are functioning correctly. And so there's a portion of plant nutrition that's just overall plant health, keeping the plant healthy. When it's healthy, it's going to be better. It's going to have better resistance to pests and disease. It's going to better able to defend itself through the use of biochemical markers and production of like phenols and terpenes and all these defenses. These natural defenses that the plant can deploy against pests and diseases. And so when you look at terms of pest and disease or IPM and which are the key defenses that the plant uses, there's a couple. So we look at like just the dermal tissue. You can think of that almost as kind of like a large gate or a large wall around a fort. It's just there to kind of stop pathogens from entering into the cells. And so having, proper nutrition in terms of like calcium, silica, and boron is going to be important to really making sure that you have strong cell wall structures. And so that's going to help to reduce the penetrative effect of some of the pathogenic fungi, especially foliar pathogens that try to get in through the dermal tissue. And so then when you look at other things like potassium and manganese levels, or having proper potassium and manganese is going to help to enhance the plant's natural defense and natural production and release of biochemicals, right? So we're going to be talking about phenols, we're talking about esters, we're talking about chemicals that could deter certain pests, but at the same time attract pollinators, right? When we're talking in terms of other types of plants. So aromas are a way that the plant uses, are something that the plant could use as a deterrent. There's been some recent information that came out, I think last year, talking about how limonene is a deterrent to whitefly. So we look at how the plant can protect itself that way. But at the same time, ensuring that you have proper nutrition levels, right? That you have enough is important, but you also don't want to have excess, right? And we talk about susceptibility of the plant to attack. We want to ensure that we're not, creating a condition so where the plant becomes too much of a target, right? So in terms of like nitrogen, so even though nitrogen is a key component in plant growth, in excess, nitrogen can create extra soft tissue, especially in the growing tips. And the extra soft tissue is going to be basically a beacon for pest and disease, right? Because it's fat, it's plump, it's juicy, it's soft tissue, it's easy to penetrate. And so they're going to go after those parts first, especially like mites and aphids, where there's a chewing or a sucking type of an insect. But then when we start to look at, on the flip side of that, we say, okay, you want to make sure you don't have excess nutrition, but you also want to make sure that you're not deficient in nutrition, right? Because that's going to decrease the plant's ability to really protect itself, right? To enact those biochemical defenses. And then also at the same time, as you have a deficiency, the color of your leaves might change from a green to yellow, and that can also be an attractant to certain pests and diseases. So, pests are going to search out the weak ones in the garden. They're going to attack there first. They're going to establish there first. So by just being able to make sure that we're to have proper nutrition, we're able to have a healthy growing plant that can protect itself first. That's always going to be the first line of defense. And that's really where nutrition comes into play, right? Just making sure that the plant is stress-free, that its natural processes are able to take place, and that their natural defenses are able to take place.
Amazing. Appreciate that, Damien. And thanks for jumping into this this topic, because obviously when we were talking before the show, it's going to correlate directly into how we manage pests and diseases. And something that came up that Matt pointed out was that not all insects are pests. And so, Matt, I would love for you to touch on a little bit about that and really what it's doing to help us create a balance of, when we're talking about inputs and what these, pest identification challenges that some of us may come across, but really how does that correlate into plant health and nutrition?
Well, I think it's really important that, we take a step back and we consider even just the terminology that we're using, right? I think for a lot of people, they see any kind of like bug looking thing, any kind of insect or anything that looks out of place, and they immediately think, oh, this shouldn't be here. This isn't right. This could be a threat. Or rather, that's giving too much credit. For a lot of people, it is a threat. It must be a threat. It must be some sort of a problem. But that's not necessarily true. And actually, on my YouTube channel, my most popular video is about mold mites and predatory mites, and people see them in the soil all the time, and they freak out and they think, oh, this is a huge threat, and they spend lots of time and energy and labor and all this stuff treating a phantom problem. When if they knew a little bit about the environment or the ecosystem, or knowing a little bit about what they could possibly see, what is a pest, what's likely to be a pest for the crop, and what isn't, they could kind of avoid that potential issue or that phantom problem. So it's all about problem solving in that way. And a big part of that is like knowing, like I said, what is going to be a threat, having a list of what these pests could be, and also pathogens for that matter, right? Fungi, bacteria, and things like that, what their symptoms are. And sometimes they don't have symptoms, and you have to use certain techniques or tools to verify. So it's not always straightforward either.
That's a really good point that you brought up. It's not so straightforward, which is why, Ajit, I think you and I have had this discussion plenty of times, right? Early surveying and detection, right? How do you incorporate a strong testing protocol in your farm when when obviously it's very difficult to see. So talk a little bit about that, Ajit, and what you're thinking around early detection and surveying.
Interesting point that Sergey you pointed out is understanding when a plant is diseased, right? Or when there is a pest pressure. And one has to understand that from looking at the diseases. And everybody knows who has worked in the crop or any of the plant health cultivation practices, the plant diseases represent a significant problem. It's all about managing and controlling those diseases. It's not about eradicating. It's very difficult to do that. It's not going to be cost effective. Now, how do you actually control the diseases? First, obviously, you need to know what is the pathogen and what is the vector for the transmission of the pathogen. So we can classify these diseases broadly into diseases which fall in caused by fungi, viruses, viroids, bacteria, then you have insects and nematodes, and there can be a broad spectrum of things that are that can be causing diseases in plants. And then the interesting point is when I look at cannabis and I look at the diseases, I mean, you can broadly classify them as diseases which are affecting roots. Now, these could be your fusarium, your pythium, and nematodes. Then you have diseases of the stem. That's your stem canker. You have the root rot, the crown rot. Then you have diseases which are more systemically in the plants. And these are more of the diseases like your viruses, viroid, and as well as the vascular wilt. Then you have foliage diseases, which is your powdery mildew, your blights. Then you have diseases that are caused by mites and aphids, which are the herbivory sort of diseases that we can talk about. And then you have disease of the flowers and buds, or the buds, which is most of the cases, it's the mold that really cause significant loss at the end product, right? Now, coming back to the question about Sergio, these are a vast different types of diseases that the plant can manifest. But one has to understand not all these diseases are visually visible. Many of them can be cryptic and dormant for a long period, and it may go unnoticed. And the classical example for them would be fungi and viroids. Now, the fungi can be dormant for a long period before it sporulates and then spreads across the whole cultivation facility or grow area. Then you have the viroid case, which we have discussed a lot about HLVD, the hop latent viroid. This can be dormant. It's not systemic. It can be certain parts of the plant. It's not easy to have them detected. The plant may grow normal until you start seeing the denting phenotype or you see the brittle stems and the leaves. So many of these diseases don't manifest it as a phenotype, and therefore you need to start to apply some of these diagnostic tools. And the best way to prevent diseases is to be able to identify those diseases pretty early in the facility before it becomes a full-blown and out of control. And so what we do suggest is to use these highly reliable, cost-effective, sensitive molecular tools. Maria, you might have a slide there which talks about the different tools that can be used. These are all DNA, RNA-based tools. They allow us to be able to detect specific pathogen because we can use the computational biology or the sequence information to identify specific fusarium, specific pythium, specific botrytis, and be able to nail down those disease at a point when they are still inside the plant, not expressing itself. And that is what gives you the the benefit of being able to use these highly sensitive molecular tools to be able to do the early detection. And once the early detection is done, then you basically have to put in a rigid protocol or SOP that allows you to continuously monitor it. The idea here is to bring the disease to an economically viable situation where it doesn't cause a huge crop loss, and we are able to manage the disease within the facility. And so here is the different molecular tools that I was talking about, and these are all the tools that classically is used. That are more over there, but what I wanted to give the audience is an idea about these different RNA and DNA based detection methods. They have their own pros and cons, and the reason we think that you have to have these tools in hand is you are trying to control the progress of the disease and before it's full blown in the facility. And I'll talk about some of the tools that we have also recently brought into the market, and we have been doing the diagnostics with that, especially for HLVD, which is now also possibly which we think we can now apply for other diseases also, for the viruses, fungi, etc. We'll talk about that. But going back to Sergio's question about how do you manage diseases, we do need to have a thorough understanding about the disease, the epidemiology, what is the causal agent, and how do you manage those diseases in case you're not able to eradicate it.
And I would love to bring up one point, kind of going back to insects and pests and some of these things that could be a potential plant vector or a vector for disease. And Matt, I think you've been touching a lot on the root aphids. Aphids in general have been crippling facilities over the years. And, a lot of these plant pathogens definitely are, I think they've been proven that they can be vectors. And so how do we incorporate, some of that knowledge and education that we've recently discovered around transferring vectors and plant management with overall disease control?
I mean, I'll take that one. Is Maria here to put up some of the pictures that I loaded?
Yep. Billy, were you going to be able to pull up one of those slides? Awesome. Looks like it's loading.
I have a few different pictures actually. So the one that I'm looking for, let's go. Yeah, go leftwards. Yeah, here. This is the one. No, that one. Yeah. So this is from a research report. Unfortunately, I just I took a bunch of screenshots from a big, huge everything you want to know about aphid video that I'm going to be publishing soon. And one of the cool things, or not so cool things about aphids, is that so they're born pregnant. When they're born, they're already pregnant with the next generation. And so they can switch between asexual reproduction and sexual reproduction. This is called telescoping generations. But what's fascinating is that recently it was found that the aphid mothers they'll take in the inputs of the environment. So like photoperiodicity. So when the light is on and when the light's not on. It'll take in other factors like gustatory cues when it's feeding and that sort of thing. And it takes that information and then it puts that information into its offspring so it can prime its offspring on the host. And that's one reason why aphids are so dang good at breaking things like plant resistance. So some plants might be naturally resistant, but then given a few generations, they can bounce back and then adapt to them. Aphids are really good at this. Most aphids are actually specialists, and so they get to be really good on one or a few different species, and that kind of continues. But others, like the rice root aphid that Sergio mentioned, is a generalist, and so that implies that they have this massive gamut of hosts, and in order for them to actually feed on them and do well, they have to have a really versatile physiology in order to do that. And so this is just one example of what aphids can do to that end. So yeah, as you can see in the diagram, there's a depiction of these so-called maternal signals. Those are those blue dots, and those are moving into the embryos, the daughter embryos of the aphid. They're actually born pregnant twice over, so not just maternally, but grandmaternally. So they're already grandmothers when they're born, which is pretty amazing.
Which is also incredibly frightening, right? That's why they're so hard to manage. And full eradication takes a lot of work, takes a lot of cultural practices and behaviors. And Kevin, I would love to touch on this with you, man. Like you're managing 1,400 lights in a commercial double stacked environment. Pest pressure is something that you've been very accustomed to working with, and like Matt was talking about, some of these things that just are, they're always going to be reproducing. Talk to us a little bit about how you've been able to manage in your facility there.
Yes, thank you, Sergio. Yeah, it's quite a challenge when you have so many plants in a big facility. We operate a high plant count, so not only that many lights, but we have a lot of plants, typically around 3,000 per room. And so managing that, particularly when you start a facility and you don't have any plants, you ask a nursery for 3,000 plants, you're likely to get plants with bugs or pests or pathogens. So we jumped straight into identification, how to manage those pests and pathogens. So it's, and with cost, such a big factor, not every option is on the table. So for me, it wasn't so much I can't turn away 3,000 plants. It's okay. How do we deal with it? So in the facility, we employ a lot of plant dips at almost every stage, and that minimizes the number of touch points because we have to remove plants at different stages. So every time we move them, we dip them, and it helps eradicate your pests that you could have. Obviously, not all pathogens. So then it goes into identification. So the pests get past a one set of dip or two sets. We educate our pruners and all staff members. I educate them on identifying pests and how and different cues, and not only that, but deficiencies. All plant health. So that way, we can catch it early. So education is a really big part of the spread of pests, and making sure your employees and everybody that is involved in the cultivation, it understands the need to control it and also identify it.
And Kev, with seeing so many plants come in and out of that facility, let's talk genetics, right? How big of a role does genetics play with this, and how, nutrition and overall plant health optimization kind of ties into that? Do you see a correlation?
Yeah, definitely. Had thousands or so of different genetics in here, bred in house, pulled in the beginning, and it's wild to, like Matthew said, you'll see some genetics that are resistant to, I've had some that, that aphids just tend to not like, when we're running, 10 different cultivars in a room, and they gravitate towards one specific variety. But, breeding for the, breeding to for that is a challenge in itself. In a commercial facility, we definitely tend to select for resistance, but with the cannabis market the way it is, other qualities are also selected for. So sometimes that's not top on the list, as you know.
Out of curiosity, Kev, you touched on certain pests are attracted to certain genetics, or maybe they avoid certain genetics. Have you seen a correlation with that, like from a terpene level? Is it terpinolene dominance, limonene dominance that they're avoiding or attracting? Just as an example.
From my experience, I would say more of a limonene dominant, but one particular variety I've had in the facility, we had HVAC issues and had some powdery mildew, and this one specific variety absolutely did not get it, and it was obvious it was that variety, and I kept it around and bred with it, so that one was more of a beta caryophyllene dominant, definitely not a limonene dominant variety, but obviously that's not a pest, so not an insect pest. So
Sure. And thank you for answering that, Kev. And Damien, what have you seen, along the ways of, certain genetics and their susceptibility with, certain nutrient regimens or programs that may improve the cell wall structure of those genetics? Would you like to touch on that?
Yeah, I mean, I think, when we look at terms of like the cannabis genetics, right? And we look at it in terms of where the rest of horticulture is in terms of the tools and how they do the breeding in their genetic work. We're still at the very beginning of kind of understanding what all of the genes do for cannabis, right? I mean, I think we're still looking for the coding for powdery mildew resistance. We're still looking for the coding for, viral, some type of resistance against viral symptoms. And so I think we're going to be able to answer a lot of these questions, in the next 5 to 10 years or so, coming out with, cultivars that are going to be a little bit more disease, have some good disease suppression in them, whether it be powdery mildew, whether it be virus or bacteria, or, just bud rot, things like that. So again, I think there are ways to create. So when you look at like how susceptible a plant can be to a foliar pathogen, like a foliar fungal pathogen, right? They have to the mycelia has to physically penetrate into the dermal tissue of the leaf and of the stems. And so those strains that have a high fiber content or have a high dry matter content are going to be better against foliar pathogens. And that's why things like silica is important, because silica happens to increase the plant's production of like dry matter and getting really strong cell structures around the dermal tissue. And that just helps to kind of fortify the fort to not allow those those enemies to even penetrate into it, to even establish. So being able to look at what strains naturally have some of these thicker cell structures, have a thicker dermal tissue, and you can tell a lot of times when you look at strains, you'll see that the leaf tissue tends to be a little bit thicker. Some tend to be a little bit more juicy and like lime green, light green versus a darker blue green type thing. And you just want to be able to match that with a good feeding regimen so that you're promoting proper plant health, but not necessarily promoting over lush growth or that softer tissue, right? You want to have that hard tissue, that more dry matter in the leaf and in the stems.
Amazing. Thank you, Damien, for that feedback. That's, you touched on a lot of great points there. And, one of those things that is actually tying into this question that we have from somebody from LinkedIn, which I appreciate the questions coming in, guys. And also just want to drop like good questions are going to be receiving a free HLV pathogen screening, compliments of my flora. So if you want to ask something, feel free to chime in on any of the platforms that you're currently watching this on. And let's go ahead and actually let's ask this. Matt, I'm going to lean into you. What types of pests are typical vectors for pathogen spreading that you may see?
There are several insects that will vector specifically viruses usually. And the biggest one, the one that most people know in agriculture, are aphids. And that's because they basically have like a hypodermic needle for a mouth. So in fact, they're so nuanced and they're so precise with how they penetrate the plant body that they can take they'll take little taste tests as they move along. They have a stylet that punctures through, and then they have mandibles that go through there. And maybe I don't really have a picture for this, but perhaps the one where we have a purple aphid, that middle one might be a good one. Yeah, so you can kind of see the stylet there, and they will even take taste tests of the plant cells without rupturing them, so without violating their integrity, which is great because they also will suppress the immune system, which is terrible for us when they're doing this. So they secrete saliva. The saliva has a bunch of what are called factors or compounds, and those compounds will variously disrupt the local immune system. And like we said already, they reproduce really rapidly. So you have this stacking effect that happens where one aphid becomes 5, becomes 15, becomes 30, and all kind of congregated in the same place. Well, that localized immune suppression is going to be super great for any viruses that will be passed by them. And there's various more details about this. Different aphids can or can't vector certain viruses. You might think, well, if they can take up the virus, can't they just transfer? Isn't just basically the same, but actually it's not. And a lot of research has been dedicated to finding the nuanced differences. For example, rice root aphid, it can vector in other plants at least, and I think also in cannabis, cucumber mosaic virus, for example. I think that is one of the ones that my flora DNA looks at, right? So that would be one of those things you want to look for. And there are other viruses too, but they might not be relevant for example, cannabis, but then other plants as well. So aphids have this capability, and they reproduce very quickly. And once they start feeding, they can take it up, and then they can move to the plant, sometimes great distances. Other insects, though, are like thrips. There's a lot of thrips that will be vectors of viruses that we know in agriculture. The biggest one probably is western flower thrips. And then also a relative of aphids would be whitefly. Silverleaf whitefly was the one that I have the most experience with. But there are other whitefly vectors of viruses too. No, I don't have a whitefly picture, but yeah. Well, there are great examples, though. A lot of these insects are very closely related too, and you'll notice that they tend to all have this kind of sucking mouth part. Leafhoppers are like this. So yeah, so if you can tell that an insect has this kind of mouth part, you might be more incentivized to maybe take it more seriously as a biosecurity risk rather than one that might have a chewing mouth part, although those two also can vector viruses sometimes.
Awesome feedback, Matt. Very, very informative there. And, one of the things I want to piggyback onto that is just cultural practices and tools and proper sanitation of those tools in the environment. Maria, if you can please pull up the scissor wall picture real quick, I would love to just briefly touch on sanitation protocol and make sure that we are all fully aware of how to properly clean your tools. So, I know that the white paper that Darkheart put out a few years ago on 2% Virkon being a phenomenal tool to actually sanitize and sterilize any sort of viral load presence on your shears is what I've been using for the past few years, and also incorporating some hypochlorous acid and sodium hydroxide as just 2 extra layers of protection. So part of the process, Maria, I'm not sure if you need me to resend the image, but I'll get that up momentarily. So let's just briefly touch on this. So 30 to 60 seconds of fully soaking and agitating. We like to use dish brushes, and the purpose of that is just think about brushing your teeth, right? If you just dunk your tips into the solution, whether it's 15 to 20% bleach or 2% Virkon, then you're just kind of leaving it there. I'm a huge fan of agitation, so making sure that all of your shears go fully submerged into we'll call them like a little hotel pan, right? So making sure that your tools are fully submerged and agitating, right? That dish brush agitation is key. And so as we start to go through the process of utilizing, non-porous cutting boards and, stainless steel dipping trays or hormone trays or hormone dipping stations. Thank you, Maria. This is perfect. So what you'll see here is our sanitation and sterilization section in the propagation space. So you'll see 3 different stages of sodium hydroxide, hypochlorous acid, and Virkon. This is like a nuclear treatment center for any sort of plant pathogen that could potentially come in contact with those sets of shears or cutting boards. So as you can see on the wall behind there, those are individual cubbies. Each one of those cubbies represents a brand new set of shears for each mom, right? These shears are not shared with another mom. So as you start to go through and understand your own protocol for your internal propagation, it's important to understand cultural practices and behaviors, right? You don't touch raw chicken and then go, touch cooked food. Like it's the same concept, plant juice, right? We don't want to cross contaminate. So thank you for pulling this up, Maria. This is just a clear vision just to make sure that we're not using isopropyl alcohol to try to sterilize our shears to protect from plant pathogens, especially viroids like Hopslatin viroid. So, Jose, I'm going to thank you for submitting your question here, and let's go ahead and read this. How often should one budget for testing for viruses and for plant nutrition throughout a plant growth life cycle? Ajith, I'd love to lean into you for this one.
Well, thank you, Jose, for bringing up this question. Very interesting question. And the way I would approach to that is basically, I would say that you want to have certified seeds or certified clones when you bring material into your grow facility. So the earliest time point where I would test any of these material for any viruses, viroids, or any of the other plant diseases that we talked about, and we offer you an array of different diagnostics for that, is to test the plants when they get in or enter your facility. This is where you can actually know what population of your plants are actually infected with the viroid, virus, or a disease, which again, I said the fungus, bacteria, or the nematodes. We don't have a nematode right now. But that's the point where you can actually control and manage your disease. After you know your population has a certain percentage of infected plants, you're better off destroying those plants rather than even keeping them. In the meanwhile, keep all the other plants in a quarantine until the results are back to you. Once you have those plants which have been identified as pest and disease free, then you use those materials for your propagation. At that point, it is a constant and systematic way of continuously monitoring and taking a certain subpopulation over a period of 3 weeks, 4 weeks. For example, in case of HLVD, we say that we can detect HLVD within the 4th week or 5th week post-transplantation or making cuttings. So if we can put a regime that continuously monitors, then we can bring the pressure of the viruses, viroids significantly lower, which is we're talking about keeping it within the single digits. And we have showcased this with some of our big customers that we can actually do this by just continuously having a rigid regime of constantly monitoring your plants for the viroids.
That's excellent feedback, Ajit. And one of the things I want to touch on is the lifespan of a mother, right? How long is a good lifespan for a mom? And from a propagation perspective, I like to say 3 to 4 months. And ultimately, it's strain dependent, but really, we want to make sure that we do bring in and start with healthy material. So a Gen Zero TC mother plant is a good starting point for your internal propagation. And one thing I do want to touch on as well is the overall, like, what does a quarantine period look like? And I'm going to lean into Kevin and Damien on this one, because if you're bringing in external genetics, right, you're on a timeline. You really got to lean into how fast you can get your quarantine process done, but also make sure that it's accurate. So talk to us a little bit about that, guys.
Sergio, if you can jump in for 1 minute before we switch to.
Apologies, one more time, Ajit.
Can I jump in, Joe? I wanted to just go over the MFT deck for a quick
Yeah.
grasp on
Oh,
how we
please.
actually do that. So I know I just gave you a quick idea about how you want to start monitoring your plants. So one of the recent tools that we launched in the market is MF Detect. And MF Detect is a very, very sensitive, reliable, and cost-effective method for detecting viroids. So what it basically does is it combines two different highly sensitive molecular diagnostic tools, the LAMP and PCR, for enhancing the sensitivity. And we use a fluorescent dye marker to be able to visualize it. So people who want to actually use this can actually be able to do it in-house in case if they don't want to use us as a provider. And we are trying to bring that on-site capabilities built into it. Now, the scale at which we can do is like we can do almost like 3,000 plus samples in over a day with the current number of people that we have at workforce, right? We can scale it up if in case we want to do it, but we can analyze more than 3,000 samples on a daily basis, and our turnover period is 24 to 48 hours. What we have done is we developed this tool for HLVD, and we demonstrated that actually this diagnostic tool works for detecting the presence of the viroid from any part of the plant, whether it's leaf, whether it's root, whether it's petiole. All these plant parts are sensitive. But however, we found that the leaf is more sensitive in early detection. And the other factor is that now this whole technology has been, in fact, incorporated where we now give you a patho X or X, where there are 7 different pathogens we can test using the same detection method, but it uses a different set of molecular primers that we call that is unique and specific for the pathogen we are using for. And so with this tool, now you can not only detect the HLVD, we can also detect some viruses, BCT, VCC, MV, AMV, etc. And then we can also use for detecting some of the fungi, which is very common, which is including the Fusarium, Pythium, and Botrytis. But we're expanding that. We'll have a pan genomes that we can actually use for multiple Fusarium. But this is a unique tool that is available, which is very cost effective, very reliable, very robust, and we can constantly provide you an updated data if you decide to work with us over a rigid regime, and we can make it very cost effective for you folks who are interested in using these molecular diagnostic tools.
Amazing. And you touched on 24 to 48 hours on how long it would take to receive your pathogen screening results back once you're utilizing the MyFlora Detect.
It's the same thing for most of the detection process. It is only comes down to the number of samples we are handling on a given day at the facility. But for all the diagnostics that we have, it is a rapid turnover between 24 to 48 hours.
Amazing. Which goes right in line with like a quarantine period, right? You should segregate your external clones coming in from another nursery in a specific area, so you can make sure that you're controlling any sort of pest or pathogen that may be introduced, like Kevin touched on earlier in his comment about bringing in 3,000 plants from an external source. So very, very good to know. Thank you, Ajit. And as as more tools and solutions, like you mentioned, like I believe the virus and viroid panel has 6 or 7 different viruses and viroids on that panel. Is that correct?
Yeah, we have 5 different viruses right now, and a couple of fungi that we have in the current panel, but it's expanding. We constantly learn from many of the folks who are in academic institutes, from the growers and cultivators. They talk about different viruses they see. So obviously it takes time to develop the tool. So right now it's limited to 7, but it's soon going to be expanding here. Can you expect between 10 to 12?
Got it. And so awesome options, right? So you clearly can, if we don't have the resources or the space allocated to have our own internal laboratory for on-site pathogen screening, we just work directly with the lab. But if you do have a space, MyFlora does offer a service to actually provide a on-site service for pathogen screening. Is that what I heard?
Yes, we are rapidly expanding that capabilities. So we have some few clients who are working with us, and we hopefully will learn from them and expand it to others in the near future.
Cool. Good to know. If I can circle back to just a quick overview of what does a good intake protocol look like from a commercial perspective. Damien or Kevin, do you guys want to jump on that real quick?
Kevin, you wanna jump in?
Sure. So like I said earlier, with the high plant count, it's a bit of a challenge. And depending on the time of year and sourcing genetics, obviously when you have an outdoor season, a little easier to source 3,000. From my experience, you get to late summer, might be a little more of a challenge. So sometimes you feel like you're stuck with those plants. So you end up having a more of a reactionary protocol than a proactive. And to quarantine that many plants is very difficult. So the quarantine period is very short because they obviously have to be housed and be properly taken care of. So, I usually am visually inspecting as they come in and identifying anything that I can immediately. And then you could kind of put a relative percentage on the pathogen pest and assess from there. And obviously, if it's horrible, it's probably worth sending back. So that's kind of how I start the process. And then from there, you assess, oh, do they have mites? Do they have aphids? So that you kind of go from there, and you have a general dip or treatment, or you have one that's isolated for a certain pest that I know it works better. So that's usually my process in this with the high plant count. However, with that, it's such a challenge that it's much easier to grow in-house genetics, which is what we do. I haven't had any real plant intake in probably 2 years. So everything is grown by seed, and generated internally, bred internally. And that's helped solve a lot of those problems as well.
Love it.
And I would like to add to that a little bit and say, along those same lines of just ensuring that the starting material that you're receiving is clean. So I would take a step back and even say, really do some due diligence on the propagation nurseries that you're utilizing to get these genetics from, right? So ensure that they have proper testing procedures in place, that they're testing their mom stock, right? Check to make sure that they have good IPM practices in place. So a lot of it can be solved by having a process-driven approach to where you have SOPs in place that will find these challenges and mitigate them before they cause any economic loss. So I would say, lean on your nursery provider to say, hey, you need to have these systems in place to ensure that the material that I'm getting from you, that I'm paying top dollar for, that is clean, right? But even having said that, as a level of insurance, you should always have an intake quarantine area in your farm, preferably off-site, because again, a lot of these insects are small. They get around, they go through cracks in the door, and sometimes they can get on the actual employees and transfer that way. So I would have a segregated system where the employees even if for that day they stay in that area, maybe they can go and do other work in the tomorrow or the day after, as long as that they, take the, change their clothes and get into disrobe and put on a uniform, things like that. So biosecurity measures are very important to have as well. So I would even say, just lean on the nursery providers to do a better job as well. In that, and any layers that you can put in place to help protect yourself against the attack from pest and disease is going to be to your benefit, right? Because we've all seen those rooms where we walked in and 90% of the room is infected with hop latent virus, and you're like, oh my gosh, there's nothing left. And so it's very terrifying for that. It's very destructive for the grower, causes a lot of stress. So we just need to be smarter about how we intake our genetics to protect ourselves.
It's a great point.
I also
Having that
want
strong
to.
relation. Oh, go ahead.
Oh,
bud.
sorry. I just want to interject because I thought that a really good point was made. Not only should you have procedures and be very assiduous in how you interrogate the people who are providing you this stuff, like, are they clean? How do I know this? What can you do to show me? Don't just roll over for no data. And on top of that, the same thing with like plant resistance. Like if someone is saying buy my seed or get my cultivars or my cuttings because they are resistant to this environment, this pest, or whatever, interrogate that. Plenty of people I've worked with, they just say, oh, well, he said that they were resistant, so they were, and they weren't. And I understand that right now that's a little bit difficult to assess, but that's becoming more and more possible. But it's one of those things where, like, if people are asking to pay a premium price for premium product, then try to find out whether that is actually the case. Do your due diligence. I definitely want to emphasize that point. It's a great point to make.
Couldn't agree more, man. Trust
And
but
may I
verify.
interject here also as well, Matt? And Matt brought a good point here, which is going back to the genetics. It's not only the material that you get, which needs to be confirmed as clean, but also make sure you do the due diligence on knowing the genetics of what you're getting. And I think we do have a service for that, which is based on a DNA technology, which assures that what you've been told is what you are getting.
Yep. Which was, thank you for bringing that up, Ajit, because, one of the things that we recently did at the Connect community was host the LCG Battle Royale in Los Angeles. And Damien, thanks for being a part of that. That was a fun event, and kind of just touching on genetics. And, ultimately, like, we've seen so many different LCG cuts floating around the marketplace. It's a great cultivar. It's a great flavor profile. It's in demand, but let's actually use a genetic fingerprint to identify and compare, from the, just from the overall identification perspective. It was really cool to see all of the 11 entries that were participants in there. And by the way, every single one of them was so fire. Excellent quality grown flower. And we're going to be doing another battle royale coming up here in the near future in Las Vegas during BizCon week. November 30th, we'll be putting out a little bit more information on that coming up soon, which is going to be powered by My Flora DNA. So we want to utilize this trait agnostic fingerprint service to be able to help us identify the genetic similarities between each of these. And, again, I mentioned trait agnostic for a reason, right? You can't do anything with these markers. It's less than 3,000 steps on that fingerprint. So just be cognizant of that. We want to be able to use this as an identification tool, which, again, there's plenty of use cases that we'll be exploring and covering on that. But again, just from like a genetic validation perspective, really cool stuff coming out there. And again, really stoked to be a part of this with you guys. Now, we've had a lot of great questions submitted here. Let's go to Carly Bader's question here. Are there any kind of tent or separations you would recommend for cultivations that don't have any specific clean spaces? I'd like to take that.
I mean, anything, really anything that you can do to just create some type of physical separation. And we've been talking a lot about large scale commercial operations, right? And one of the things that we've learned is that each grower has a different tool set to kind of go after the challenges or to solve the challenges that they have in front of them. A lot of them have the same challenges and they find different ways to overcome it. So, anything you can do to kind of just create a simple separation, whether it's a grow tent or a separate room, maybe put some sticky cards up around it, do some kind of exclusionary things that you can help to just add extra layers of protection there to make sure that if a bug does happen to get out of the tent, then it's attracted to that sticky card and then it gets stuck on the sticky card. I mean, anything you can do in that regard to just keep them separated is going to be better. If you're working those plants, don't go and work in your normal garden. Like, just stop working for the day because you don't want to transmit any type of pest or disease that could be on your body.
Awesome feedback. Thank you, Damien. Appreciate the question, Carly. And let's see here. Let's go back to Andre. Andre asks, how does the use of beneficial microorganisms and soil amendments contribute to the success of ICM practices? I'd like to take that.
Yeah, I'll take that. A lot, right? But it can be. I want to say this point first up front. It can be really hard to assess that in the field, right? I think that over time I've become pretty allergic to like broad generalizations that people will make, especially people who are trying to sell a certain product or a service or something like this. And I get it; it's my own work as well to assess these sorts of things. But sometimes people will just say, "Oh, it's got good microbes in it," and people will go, "Okay, cool, all right, that must be enough." But that doesn't not necessarily the case. That can be a very very fuzzy, very lossy statement to make. But research definitely shows that there are all kinds of microbiota that can have beneficial and detrimental effects. So knowing what some of those are, and some of them are commercially available, right? My favorite example of that would have to be the entomopathogenic fungi and sometimes bacteria as well that people often utilize. But what's nice about those is that they have a specific effect and a reliably simple effect, right? Beauveria bassiana or Bacillus thuringiensis, right? You apply it, it affects one or several different kinds of insects, and you kill them with it, right? So that's really obvious. Some microbes are going to, help in more discrete ways, which can be again hard to assess, right? So they might be like plant growth promoting bacteria, maybe they don't play nicely with something else that you've added or something else that's there and you didn't know it was there. And even if you were to take if you were to sequence the genome, um, rather not the genome, but like if you were to do like a sample and search it for genomes and try to find out and figure out all the different species you could that were even identifiable, that's still just a snapshot in time, right? That's just one moment in time. The population can go higher and lower, after irrigation, before irrigation, first month of growth, last month of growth, right before harvest, right? So, the general answer is yes, but be careful with like I guess that's kind of the theme that I'm bringing to this conversation. It doesn't have to be the only thing I say, but trust but verify, right, when you can. And I think also it's important that like when you're talking about soil amendments, try to trial them. Try to like actually see like, did I see a big difference if I just control for other variables? Did the plants really change all that much? Right. I think that's really times they're more in love with the aesthetic, or they're more in love with something sounding cool or sounding like something great, or somebody has lots of charisma, and that's great, but ultimately I like to bring a more empirical, analytical thrust to this kind of a thing. What do you all think about that?
Couldn't agree more, dude. I mean,
Thank
that's the key.
And
Trust but verify. And oh, I'm sorry, Damon.
Go ahead. I was waiting for you to finish. Sorry. Oh, I was just going to say that in terms of plant nutrition as well, we know that there are several species of beneficial microbes that help to enhance the plant's uptake ability. So again, like in terms of promoting proper plant health so that the plant can properly defend itself against attack, we've seen that there have been some soil microbes that help the uptake of nutrients, and also that protect the root zone. Like Matt said earlier, it's sometimes it's just the fact that one species is in that space and it doesn't allow, it just outcompetes the pathogenic species. So I mean, we do see a little bit of that happening here. But I agree, like sometimes these inoculations take time to really take effect. And cannabis is really, like in terms of commercial cropping, cannabis is a very fast crop.
Yeah, that's an excellent point, right? Because are you going to get the benefit? So I think that as research becomes more prominent, we'll kind of eke out these nuances and see, like, first of all, not just single microbial species, but different strains, and also different consortia that can be applied. And that's actually, I think, that's the really cool future. I guess it's here now, actually. It's still kind of nascent. But that's what I'm really excited for, is taking that genetic testing ability and leveraging it with what we know about the various microbes that could be almost like N of one health, if people are familiar with that topic in medicine, where every person is individually unique. And so you can kind of tailor things to people. And I think the same thing could be true for cannabis in general, and also different cultivars and different grows and contexts. So that is what I'm very excited about. I think it's very possible to do.
And so both of you have a great point over here. Maybe I can interject and bring in a little more of a broad spectrum based on the other seed industries, what they do. So we talk about biocontrol. That's a great point of view, whether it's a biofertilizer, it's a bacillus species. But the fact is also that we also talk about the soil, right? The soil microbes, the soil health, and the different cropping systems. So one of the ways you control the amount of nematodes, insects that's soil-borne is to do crop rotation, so that you engineer the soil to be able to have a diversity of microbiome biomes that naturally gives you an enhanced growth and vigor to the plant, but also controls the dominance of certain insects and pests in that given microenvironment. So that's something that is part of now the regenerative ag that we talk about a lot today. It's not about sustainability anymore. It's about regenerating our agriculture, going back to some of our traditional practices, which eclipsed during the Green Revolution. But obviously, we are seeing back to the opening where we know that we can actually monitor and control the productivity and the pests and diseases by just making sure our soil is healthy, and we have regenerative ag practices in place.
Yeah, it's been very fascinating to see sort of recursively scientific methodology to come in and see how we can take older, more traditional techniques, like you're saying, and see how they could be utilized more efficiently, with more precision. So precision agriculture meets traditional agricultural methodology. I think it's a really wonderful thing, especially when we know that domestication over time has it has given us some great benefits, but without us knowing, in the millennia that we've been growing plants in agriculture, sometimes we sort of make trade-offs, and we would like to reintegrate things like defenses and certain microbes that maybe were lost from the origin point where the wild ancestor of a crop is from, or something like this. So that's why I find like evolutionary research really appealing, and eco agroforestry and that kind of thing. So I'm glad that you bring that up. That's something that I think is more possible than people realize, but it does require some work and some thinking. But farmers are some of the most creative people I've ever known. So the ingenuity is there.
Exactly.
And to add on to all these is the fact that we still talk about not only the rich soil environment, but also the fact that you reintroduce the facts of bringing the diagnostics, trying to monitor the soil health by just doing diagnostics, but then also bringing the diagnostics pretty early in terms of controlling diseases, whether it's an outdoor facility or it's an indoor facility. The fact is, the disease pressure varies by each of these facilities, right? Obviously, natural predators or biocontrols or factors like which are competitors or antagonistics. Those are all going to be able to help you out when you are in an outdoor facility. While coming into indoor, you have a lot more factors that you control. And that's what the molecular diagnostics comes very much into practice. And you have to have a rigid regime on that. And the fact that we can tell you that some of these diseases can be monitored pretty much early in the introduction stage when they are brought in, as we say, as mother plants, you control practically lots of diseases and pests pretty early in the process. Then you basically have to build an SOP, which basically goes back to this HLVD case where we said, hey, every 3-4 weeks, if you start monitoring, then you're going to be able to monitor and control the population, the pressure that is coming from the HLVD infested plants. So you significantly reduce that by just doing these simple switches in our cultural management practices, which is based on a highly rigid regime of how you monitor your plants, how continuously you physically and as well as use some of these sensory tools, incorporate them into your management practices, and then you see a significant value out of it. It seems to be upfront a cost, but you will see the output of that, which is going to be beneficial for you.
Amazing. This has been awesome, guys. I want to start to circle this back around as we come to our closure. And something that we touched on earlier is we're each going to give each going to get an opportunity to kind of give our key takeaways as we start to wrap this up, so we can have some good notes to walk away with and just key considerations. And I'll go and start us off. My biggest one that I've been preaching for a few years is really about cleanliness and clean behaviors. Right? Be sure you're properly sanitizing and sterilizing your tools. Culturally align your team in the cultivation. Understand what is a plant vector or a pathogen vector and pests identification. Let's celebrate these things, right? Let's make sure that people are recognized for their effort to care about what's going on in the farm, right? Identification of pests and, anything funky looking out, they're utilizing some diagnostic tools, like Ajit, you just mentioned there, right? Like, we have science at our disposal here. Let's use it. And as the, leaf tissue analysis and soil analysis companies that are out there in conjunction with pathogen screening, these are things that we can utilize to make sure that we are growing a healthy plant and bringing out the best genetic potential of our plants too. So that's me. I'm going to go ahead and pass it along to Damien, if you'd like to start us off. Damien, I think you might be on mute. Or is that me?
Sorry about that. I was just saying
that,
yeah, in terms of like plant nutrition and IPM, I think that, a key takeaway is just ensure that your plants are going to be well fed, right? Because if it's well fed and it gets what it needs, it's going to have the best chance at defending itself. And that's always going to be the first line of defense, is allowing the plant's natural defenses to kick in first before you decide on doing any other type of chemical or any other type of controls. So yeah, happy plants or healthy plants are happy plants.
That's it. They feed off our energy. Stay positive. Have gratitude for this beautiful plant. Thank you, Damien. Matt, how about yourself?
So I think that the key takeaway, at least from an IPM perspective, or IPDM or ICM perspective, is that basically you want to know, step 0 is know what problems you could have are. So know the pests that you might be able to encounter in the crop, in this case, right? Cannabis. But this is true for any crop, really. Know what they look like, know what their symptoms are, know what kind of damage they have, and what kind of threat level. Like, are they a huge problem, or are they kind of a low threat sort of a thing? And then have options already available to you, not just knowing what some solutions would be for your particular context, but also what would you, or what would have those options actually on site, so that you're not scrambling around trying to look for a solution. Obviously, there's like hundreds of different possibilities in a lot of cases, but the major ones you certainly have a plan for. So that's why having an SOP, an and IPM SOP is so important. And then on top of that, none of this really matters if you're not able to actualize your plan. So part of that plan is also scouting your crops, looking for potential problems. We mentioned a quarantine already today. That's really important. A lot of people I work with or I've worked with didn't really do that, and I think they could have benefited greatly from that. But even things can get through quarantine. Not everything is visually obvious. So crop scouting, and then also on top of that, testing is also something that people should do more often, I think, especially at a commercial scale when a potential issue can cost you quite a bit. So the prevention is, of course, less expensive than the cure.
Love it. That's a great takeaway right there. And always leaning into trust but verify. You know, that's one of the things we can all appreciate and take away. Trust but verify. And last but not least, Ajit, how about yourself?
I think you folks have touched most of the points that I want to, and I don't want to rephrase them, but I would say that, as you said, trust your genetics, make sure you have the right material, have a rigid SOP that allows you to constantly monitor your material that is inside your facility as well as that is coming through your intake process. So everything starts with the material. And I would say start with a clean material, then incorporate some of the advanced tools, keep all options open. There are many different technologies which are available, and all these technologies also provide services and solutions. So you want to keep your options open, like as Matt said. Be versatile, work with the different providers and see what best is the option that works for you. And we are in the business to provide you those solutions, because without you, we can't know about what the diseases are and what the problems that you have in your grow facilities. So it's a constant two-way communication for us. We are here to support your growth, provided we know what exactly is the problem. And then, as I said earlier, once you have set up a population where you know that pest and disease pressure is limited, it's only a practice, a very healthy cultural practice, or grow management practices that allows you to continue to monitor and keep the population pressure from diseases and pests at the lowest level, so that you can make the best out of your grow facility, and as well as be able to get the business output that you want. So I think my perspective is keep hygiene, cleanliness, and have a high reliable molecular diagnostic capability built, so that you are able to control and mitigate, if not eradicate, the disease or the pest.
Fantastic. Ajit, well said. All three of you guys, thank you so much for your key takeaways and ultimately just taking time today to educate the community and have this engaging platform. Maria, I want to thank you too for allowing us to have this space and share it under the CannaScope. Wherever you're watching this, whether it's on LinkedIn, YouTube, Grow World, shout out Nick. Thank you for having us on there as well. We look forward to hosting more events through all these platforms together. And again, guys, just opening up the community. This community is here for us to learn and leverage each other to grow. So that's ultimately what we're here for. Thank you all for tuning in. Be sure to like and subscribe on the newsletter so you stay up to date, you stay informed. We'll be having more of these discussions. Really hope you guys got some good takeaways from this. I know I certainly did. And until next time, thank you all for coming in. Appreciate you. Have a good rest of your day, guys.
Thank you, Sergio.
Thank you for
Thank
having
you
us.
all.
Cheers.